Hello everyone, so welcome to my channel. This is Shruti Oja, and here we are going to talk about biochemistry plasma proteins chapter. So with this chapter I will set an ideal template, or you can say some ideal method how to study biochemistry and how to follow different books at the same time and in a time saving manner. Since I have done all this, so I can show you an example or a template for you, like how we have to write in points, like what headings do we have to create, and what are the important questions in this chapter. So first of all, the functions of plasma proteins, electrophoresis and its applications, total protein concentration, structure and function of immunoglobulins, structure of IgM, IgG, whatever it is, what is the valency of each antibody, and functions of albumin. Acute phase proteins can come as a clinical question, and also multiple myeloma is a very like important 5-star question. So these are basically the important questions from Harper. If you will open the book as suggested, because I think that is a very nice book and you must follow that. So this is a very like good list of questions. These have some questions like of iron mixed in it. So when you study the iron metabolism also, you can solve these, because these are very conceptual and wholesome kind of questions which can cover many concepts in one go for you. So let's start the contents of the video. Plasma proteins, that first of all the types. First of all, what are plasma proteins, then types, functions, and the basic important categories of the plasma proteins. So here we are having the plasma proteins. Plasma contains hundreds of different types of proteins, right, which are known as plasma proteins. Whenever you start a question, you need to write the definition of that particular thing. If they have asked about C-reactive protein, if they have asked about albumin, fibrinogen, or plasma protein as a whole in a question, then you need to write what it is in a one line, two line, whatever it is possible, because that gives a very wonderful impact on the examiner, because we have to write the answer in a way that the teacher knows nothing. Next, normal total protein concentration is 6 to 8 gram per deciliter. This is a very important value, and this you need to remember till the end. Okay. All plasma proteins except immunoglobulins are synthesized in the liver, and except the von Willebrand factor as well. Most plasma proteins are the glycoproteins, but lipoproteins also do exist. Basically, lipoproteins may or may not be included according to different authors, but yes, they also do exist. Now, classification. Earlier it was based on something else, but now it is based on the electrophoretic pattern. So it is based on the electrophoresis. You need to write, if you are writing the classification, on what basis we have classified. It is not so you will write some classification and it is classified into albumin, globulin, fibrinogen, but how? You need to make it clear. So albumin, this value is also very important, 3.5 to 5 gram per deciliter. Globulins, fibrinogen, all these values are very important. Next is albumin. This pattern is called the densitometer scanning. This can be asked like a very small question in vivas only, but you need to draw this in theory when plasma protein part comes under the classification. So this is the serum protein electrophoresis, and this is the sites, I hope so you know that. This is albumin. All this you can do by your own, but yes, you need to learn all this, right? Next comes the types of plasma protein we have classified. Now we have to explain that. So albumin is the major plasma protein, constitute up to 60% of the plasma proteins. Human albumin has 585 amino acids. Okay. Human albumin, it is synthesized in the liver and it is a simple protein. I want to say a disclaimer that these are the notes of from the Prasad biochemistry book, plus the Rajesh Jambulkar notes text book, and plus the Harper. Next is globulins, constitute many different proteins separated into four distinct, we know, according to the bands in electrophoresis. Now, alpha 1, alpha 2, beta, and gamma. So, A is very important. A. First letter, so very important. We will divide it into alpha 1, alpha 2. Okay. Beta and gamma are not so important. So, beta and gamma. Alpha 1, antitrypsin, retinol binding protein, thyroxine binding globulin, and HDL. Just remember that the retina is somewhere present, thyroid is somewhere present, so these are in alpha 1, very very important, right? Next, alpha 2, haptoglobin, cellular plasmin, prothrombin, these are circulating things, right? Are not some not organ type of things, so they are alpha 2. So primary importance organs, so retinol, thyroxine, etc. I am just saying some mnemonics so that it is easier for you. Next is alpha 2 globulins. This is the alpha. Beta globulin is the LDL, transferrin, and hemopexin, plasminogen. Even if you learn 2 to 3 or 2 even, then that is also enough. But learn the important ones. Gamma globulin, we know immunoglobulins. Fibrinogen, that is responsible for coagulation, right? That is converted into fibrin in active form. Next is serum proteins are class separated by electrophoresis. This we already know. It can be agar gel or paper electrophoresis. If someone asks, this can be a point you can answer. Next is functions of plasma protein as a whole. So first of all, maintenance of colloid osmotic pressure. The major protein which is responsible for it can come as an MCQ, that is the albumin. Okay. Now, transport functions, various proteins are there, transferrin, the cellular plasmin, haptoglobin, hemopexin, lipoproteins. These are major or transport functions. Buffering action, mainly albumin. The points like the most, mainly, or I can say the important protein for it, or we can say the exception or not. These points, wherever these words come, you need to put your extra attention. Next is protein reserve nutritive role, which is basically the albumin. Blood clotting is fibrinogen. Defense role, immunoglobulin. Just keep in mind the proteins and just write down what do they do. We know fibrinogen, we know immunity. In immunoglobulins, gamma globin, we know, and albumin also we know everything. Okay. Clinical significance is very important whenever any part comes. If a short note comes on functions of plasma protein, first of all, you will write the definition of plasma protein. In small text, you will write the classification, and then you will write the functions, and at last you will write the clinical significance as well, because without this, your clinical biochemistry paper is incomplete, because you're not studying biochem, you are studying clinical biochem. Next is decreased total plasma protein concentration is an indicator of liver disease due to defect in synthesis, right? Liver is not well, then how will it make albumin well, right? Or nephrotic syndrome, like the synthesis is not well or the excretion is more. So nephrotic syndrome due to increase in pore size leading to abnormal excretion of albumin. Now, albumin level is also decreased in nutritional deficiencies like kwashiorkor, burns, and in disease like tropical sprue. So many times we forget this burns, okay, and eventually we lose marks. Edema is main consequence of hypoproteinemia or hypoalbuminemia. Okay, edema. Now, plasma proteins, especially albumin, maintains the colloidal osmotic pressure, that is COP, which is responsible for fluid retention between the body compartment ECF and ICF, that is extracellular and intracellular. Decreased albumin level causes fluid redistribution, water enters the tissue and causes edema. So what is the main cause of edema? It can come as a short note. In plasma protein chapter, the cause or mechanism of edema and how it is related to albumins. Okay, albumin से कैसे related है? Now albumin, it can come as a short note as well, but very low chances, but yes, you need to prepare because it is important. Albumin, major plasma protein, 60% of the plasma protein. Human albumin, I have told these things already. Now when functions come, we already know colloid osmotic pressure. Okay, of blood is COP of blood is 25 mmHg and albumin accounts for 80%. This percentage can come. Okay, transport is basically transport of hydrophobic substances. Why not hydrophilic? Very simple. Hydrophilic, water loving. If it will get dissolved in the water, it can go with the water. The substances which can't go with the water, albumin is a social worker for them. It will take them in it and help go through the water, go through the blood and reach its target destination. Next is buffering action. Buffering action, we know it can accept the or donate the protons and can regulate the pH as well. Supply of amino acids to the cell. It is itself made of like the amino 585 amino acids, so it can go to the cell, can offer himself like take me and have some amino acids, so that will also be of help. Now functions of plasma protein. Now colloidal osmotic pressure, transport function and other functions. This is an exam tip. I got it from book only. Now you can be benefited from it. If functions of albumin is asked for 5 marks, write all the functions plasma proteins except the coagulation and defense functions. Note that that means like I told you the functions of plasma proteins, write all except the defense, that immunoglobulins and fibrinogen function, write all. Okay, now note that answer looks almost similar to functions of plasma protein as albumin. Why? Now this is very conceptual. Why the answer is so similar? Because albumin constitutes 80% of the we can 60% of the plasma proteins, right? Now functions of albumin: colloid osmotic pressure, nutrition. Nutrition can be added, okay, as a amino acid function only. Buffering action and transport of free fatty acids, steroid hormones, ions, bilirubin, that is the hydrophobic, okay, phobic substances. Next comes globulins. Similar to albumin, we can have globulins as well. Now alpha 1, alpha 2, beta, gamma. It has much content in it. That is why it can be considered for a question. Functions of globulins. See, either the question is the topic is very important for you, or the topic has lot of content so that you can replicate some like 40 to 50% in the exam. That is why that topic is considered as a question. So alpha 1 antitrypsin, it inhibits all the serine proteases, because this protease is having serine at their active site, like trypsin, elastase, plasmin, thrombin, cathepsin. These are example of serine proteases. Deficiency is implicated in emphysema and certain liver diseases. It is an acute phase protein. So I will talk about acute phase protein afterwards, but these are the serine proteases. So it inhibits all the serine proteases basically. Now alpha 2 macroglobulin inhibit the protease activity, serves as anticoagulant. Haptoglobin binds with free hemoglobin. Okay, haptoglobin is also an acute phase protein. Ceruloplasmin, which has peroxidase activity, incorporation of iron into transferrin, then retinol binding protein, thyroxine binding globulin, lipoprotein, hemopexin, immunoglobulins. So all these are globulins. So you need to know something about all. Okay. Next comes, these are some additional points about alpha 1 antitrypsin, alpha 2 macroglobulin, beta lipoprotein LDL, and these are the gamma globulins. So this I am sharing in my group, this PDF basically, which can be helpful for you. Next is acute phase reactant proteins. Now what are they? The level of these proteins are basically raised in inflammatory conditions. So this, it is named so because the C polysaccharide of pneumococci is there. It is produced in liver. CRP level is increased in acute chronic inflammatory states and cancer. So all this, the things which I'm leaving, you need to read by yourself, because this is just a structure, I'm sharing it with you. C-reactive protein stimulates the complement pathway, so that is why it is related to something inflammation. Okay, now abnormal electrophoretic pattern is very important, like 5-star question. Multiple myeloma, prominent M band is there. Cirrhosis of liver, like decreased albumin, increased gamma globulin. Okay, because liver is not synthesizing albumin, but gamma globulin is being synthesized by the immune system, right? So that will increase to compensate the protein concentration. Okay. Nephrotic syndrome, I have told you, increased excretion. So these are the electrophoretic patterns. These are very important. You need to learn this. There is nothing which I have to explain. If any doubts, you can ask me as well. Now, multiple myeloma, I have told you, like M band is increased, light chains of immunoglobulins are produced in excessive amount. Now, these constitute the bench joint proteins. Excessive excretion of these bench joint proteins can damage the renal tubules as well. Now, bench joint proteins are light chains. Similarly, what that are excreted in the urine in case of multiple myeloma. Now, these precipitate on heating urine to 45 degrees Celsius and maximally at 60 degrees Celsius, but start redissolving at 60 and form a clear solution at 100. So, this way we can detect. Basically, this is a detection phenomena. Now, immunoglobulins. Immunoglobulins, this we can study very well in immunity chapter when we will study everything about immunity. So I am leaving this immunoglobulin part right now, but yes, this is included in my PDF. Okay. You just need to log on to my Telegram channel and look out for this PDF. You will get everything related to it. So this I have taken from Harper and this is from Prasad, and this structure is very important. You always get a question from it in any of your exam. You will get. Okay. Now, the question bank is here, and extra points from Harper I have included. I have underlined and made out some questions. The rest part which is given in Harper in plasma protein chapter, I will cover it in the iron metabolism chapter because it belongs to that. So I hope you are clear with plasma proteins, and if you like this lecture, if you like this video, and if it has benefited you, then do like the video and do save it in your library so that it will help you in future, and share it with your friends because sharing is caring.